Hello, my excellent biologists. I am Mrs. Sloan, and this is a video intended for AP Biology, and this is all about animal development. And if you would like, down in the descriptor of this video is a copy of the notes that my students use, two columns. One is a scaffolding of the material I will go over, and I'll help you fill it in. And column two, I would throw in pictures that help you with the learning. And then up at the top of those notes is a link to this presentation I'm giving right now. Okay, so remember to consult. The College Board gives you unit guides, objectives, and I have taken those and annotated them. And there's a link right here. It's also on my website. But I want to tell you why I'm talking about development. It's an illustrative example of two things, 6.5 and 6.6, about the regulation of gene expression and cell specialization. So the College Board wants you to understand concepts, but be able to apply it. And this is a common application. All right, so we're going to study the genetics behind development. I'm not going to go into the stages of pregnancy or human development other than to show you this change over time, what you look like at 4 days, this is before you're born, 3 weeks along in development, 5 weeks, and 4 months. And we'll talk about some of the structures in early embryonic development. So we're going to start with, I will go through the what happens and then the how it happens. But first we need to talk about how you make a baby in the first place appropriately, okay? So how do we get from here, the joining of a haploid sperm and a haploid egg, okay, to here? Literally, a baby is trillions of cells that you grow up. But the amazing thing is that you started as just one single cell. Now look at you now. You're amazing. And you've got cellular specialization. But how does that happen when you have one cell and one genetic code that made you? So that's what we will talk about. But first, in order to have an egg, it needs to ovulate. And so this is an egg getting ready to pop out of the ovary. And in the notes for my students, I made this really small. So it's not an accountability piece, but just a supportive bit of material. An egg lasts about 12 to 16 hours after it ruptures from the ovary. And that will happen about 14 days before the start of your menses or your period. Here is an actual photograph of ovulation. Okay, and for fertilization to occur, sperm need to be in the vicinity, and sperm can last 4 days. So the sperm could be there from 4 days earlier, but they actually fertilize the egg on that day as soon as the egg ovulates. So the structure of the sperm that you want to be familiar with, and this part, it goes back to enzymes, is on the head of the sperm. There's a little cap, a little beanie cap the sperm wear, and it's called an acrosome. And the acrosome is full of enzymes. And remember, there is no second place, right? It's first in wins, that's it. There is only a gold medal. And so they all will be attacking that egg. And on the outside of the egg is something called a zona pellucida, which to me sounds like a summer concert tour. Oh, I have a video here I'll show you in class. Yeah. Sperm cam. And let me keep going here. Sorry, I'll pause this. We'll see it in class. But what you can see in this top left-hand corner, let me get a pointer. Okay, so here you can see the sperm. Here's the acrosome. What the acrosome does is it starts to digest this jelly coat, and then the sperm nucleus, the cell membrane of the sperm, needs to make contact with the cell membrane of the egg, and then it can put the sperm into the egg, and these two haploids will then become a diploid zygote. And then from then on, remember, development is going to be done by what? Mitosis or meiosis? Mitosis, right? And then over time, right, you will have a multicellular organism, and we're going to talk about how that goes about happening. So here, when the sperm first penetrates it, you don't want to have another sperm get in there at the same time. Otherwise, instead of being diploid, you would be triploid. And for animals, that doesn't work out. Plants do okay with polyploidy, but animals do not. So there needs to be a way to prevent any other sperm from getting in there. So initially, there's what's called a fast block. It's like a depolarization, a wave of depolarization across the egg that prevents any other sperm from getting into that egg. And then slowly there's a slow block, and you're going to create a more firm barrier to prevent any other sperm from entering that egg. All right, so now let's start talking about what happens. So this is right after the sperm fertilizes the egg. Now at that point, you're one cell and you are a diploid zygote. Now, remember, you're going to go through mitosis at this time, making more cells. But this mitosis is very, very speedy, and there's not any time for growth. So I'm going to show you, okay, the stages of development that development is all the changes that occur in the during the life cycle. And you may want to add that on your notes right after embryonic development. So, and then just add all the changes during the life cycle. Okay, and then I'm going to go through these step by step, so hopefully you can understand it no problem. So we are going to start with the zygote, and these stages of development that we're going to go through, we're going to use a lamprey. A lamprey is a type of fish, very small. It doesn't have a lot of yolk, so it's easy to see the cells migrating around, and I will show you that. All right. So first, sperm fertilizes egg, you have a diploid zygote. It then does repeat mitosis very, very quickly without any growth. And so when it does that, it will ultimately become a solid ball of cells called a morula. So on your notes, a morula is a solid, this is the part you're filling in, a solid ball of cells as a result of repeated cell division. Okay. Now, the next thing that happens is you have this solid ball of cells, and then these cells will start to pump sodium to the center cavity, and chlorine will follow, so it's salt. So you're making it hypertonic, and remember, water must flow from the hypo. Water will start to fill this up and blow it up like a water balloon, and then what you will have right here is a blastula. So a blastula is a hollow ball of cells. So on your notes, blastula, you just got to fit in hollow ball of cells. Now, what do you call that inner cavity? It's called a blastocele. And that is the cavity inside the blastula. Now, humans are a little bit different because this is a fish, remember? So for humans, inside the blastula, it's not shown in this picture, you will have what's called an inner cell mass. And I will show you that in just a little bit. And that inner cell mass is what actually develops into the embryo. And these remaining cells out here, the blastocyst, will help develop into the placenta. Okay, so just put a pin in that, but I wanted to tell you right here, you're going to have your inner cell mass, and that will develop into the baby. That's what needs to go into your notes. Now, at this point, you think of like those red rubber playground balls that you had as a kid, and I want you to let the gas out of that red rubber playground ball. And when you let the gas out, it ends up forming two layers, an outer and an inner layer, an ectoderm and an endoderm. And when you do that, so you can see that right here, it's called a gastrula, and a gastrula is a layered ball of cells. Now, initially, you're going to only have an ecto and an endoderm layer, but a middle layer, mesoderm layer will develop. And the way that develops depends if you are a protostome or a deuterostome. And that has to do with the first opening being your mouth or your anus in that developing embryo. All right. So a gastrula, you want to have a layered ball of cells as a result of invagination. Okay, now when you make that layered ball of cells, you can see, look where your blastocele was. It's no longer there, right? It kind of got smushed between those two layers. So now you have a new cavity. Your archenteron is the new cavity, and the opening, and you can see that cavity here, the opening to that cavity is called a blastopore. Now, you and I are deuterostomes. That blastopore you're sitting on right now, it is your anus. That's what your blastopore became. Other organisms, protostomes, like mollusks or spiders, that blastopore became their mouth. So let me show you a couple of things here. Chicken. Now, when you think of a chicken, right? Cracking open an egg and you see all that yolk, that is not actually the egg. That is the food for the developing chicken. There's a small little tiny disc that you can see on top of the yolk. That's the actual egg, the zygote that's going to develop into that baby chick. So they have so much yolk. That's the other extreme. It's like lanceolate, no yolk, and then here a bird with a lot of yolk is the other extreme. So their development takes place on top of that yolk. Okay, and here that becomes important when you discuss gastrulation, because when we look at a lanceolate, it can just fold in on itself through invagination to form those layers. Here on the other extreme, the chicken, it's not going to cleave that yolk. It's just too much. It's going to happen on top of that yolk. And then intermediate here is a frog. And what happens is when they do invagination, the cells have to migrate around that yolk. See, they're working around that yolk right here when they fold in. Okay, so those are your gastrula close-ups. The way you do gastrulation will depend on the amount of yolk that you have. So now let's start talking about those layers. So remember I told you outermost layer, ectoderm, innermost layer is endoderm, and then you start to form what's called a mesoderm, which is in pink here. Across here, wherever you see pink is the mesoderm. And that will develop differently depending on if you're a protostome or a deuterostome. Now, this right here, see how it's labeled, the archenteron, that is your gut tube. Okay, that is your tube that runs down, when you think of your pharynx, the back of your throat, and then it splits and either you have your trachea or your esophagus that goes to your stomach and your small and large intestines, rectum, anus, right? Your trachea is going to your lungs. This is your gut tube right here. Right? And then moving on, here are the layers, and this is a little bit farther along in development. At this point, it's called a nerula, and I know that because here is the spinal cord, the neural tube, and I'm going to talk to you about the formation of that in just a minute. But this is a clear area to show you the three different layers and what they become. Okay, ectoderm in blue, mesoderm in pink, and then this endoderm right here is your gut tube. Now, not all organisms have a coelom, okay? Some organisms are acoelomated or pseudocoelomated. Think about flatworms. But we are coelomated. And this space right here in the mesoderm, that's the space that your organs are sitting in. Like think of your thoracic cavity and you've got your stomach and your liver and your lungs. They're sitting right here in this coelom, in this space. But scientists know right from the beginning what tissues are going to become what, and that's what I want to talk to you about. So the outermost layer, this ectoderm in blue, this is what your nervous system and your skin and like the lens of your eye will all develop from the ectoderm. The endoderm right here, your gut tube, think about going down through your mouth. There's two systems you will run into, your respiratory system and your digestive system. And that's what develops from this innermost layer, the endoderm. Everything else in your body comes from the middle layer or the mesoderm. So here I've spelled both of those out, and let's put it in our notes as well. And so as long as you know the bookends, then everything else defaults to the mesoderm. So your ectoderm is your outer layer. It will form your nervous system, your epidermis, lens of your eye. Your endoderm, skip down to that one, is the inner layer, and it will form the epithelial lining of the digestive, respiratory systems, and any associated glands with those two systems. From your mesoderm, okay, your mesoderm, I gave you everything in the notes, but it's your musculoskeletal system. Think about like in the middle of your arm, what do you have? Bones and muscles, right? That all comes from your mesoderm, as well as your excretory, cardiovascular, and reproductive systems. Don't memorize these. Just know the two bookends, endo and ecto. Those are easy to remember, and then everything else from your mesoderm. All right, now, and this is just a chart. If you want this, you can pop it in your notes, but I think you got this down. All right, so where we are right now, fertilized egg, diploid zygote, goes through cleavage very quickly, not a lot of time for growth. So here, even at the 4-cell stage and the 8-cell stage, what I want you to notice is it's about as big as the zygote. Remember your cell cycle, right? It's mitosis, G1, right? Growth, making more organelles. S stage of the cell cycle is synthesis of DNA. G2, you're making those proteins, and then you go through mitosis again. So the nuclear part, the replication of the DNA in the S stage and going through mitosis, that is all accurate, okay? That's fine. What you notice here is the G1 stage, you're just whipping right past that. You're not getting any larger at this point. And now you are a marula, a solid ball of cells. You pump in some salt, water follows, you are a blastula. And it's a hollow ball of cells. The cavity inside the blastula is a blastocele. Then you're starting to let out the gas, forming a gastrula. Initially, you have just an ectoderm and an endoderm. The blastocele goes bye-bye. Your new cavity inside of here is called your archenteron, and the opening is called the blastopore, which is what you're sitting on right now, your anus. So now we're going into the next stage, which is the nerula. The nerula is the organ level of development. And what happens is something I think fairly incredible. This is an example of an induction. And what happens is in the mesoderm, there's an area right here that I'm pointing out called the notochord. And you can see it here in this picture as well. What the notochord does is it exerts some peer pressure on the ectoderm above it and causes it to fold in. It's sending morphogens, chemical signals, and this is also an example of cell signaling because it's sending a signal to fold in. And so the cells right above the notochord will start to do just that. And that will then form your neural tube right here, which is your spinal cord. Now for us, because we are vertebrates, right? We have a backbone here. This notochord right here will become the vertebrae that protect our spinal cord. Okay, so that is an example of induction. And at this point, you're starting to develop your organs. So on your notes, you have organ stage of development when the nervous system forms from the ectoderm and the neural plate. Okay, so find your neural plate. You have a neural plate, and then eventually you will have this neural tube. And I already told you the anterior end of this, and I'll show you that in just a minute. The anterior end will become your brain, and the rest will become your spinal cord. And then on B, it's induced to form by the notochord. And they have literally done experiments where they've transplanted notochords to multiple places on, like, say, frogs or salamanders, and they've had multiple spinal cords, brain, heads form as a result of that. I'm not saying that that is a good thing to do. I'm just saying it has done, and I'm letting you know that. Okay. So next, we need to talk here. Let me get to the next picture here. This shows you that induction, and I'll revisit this a little bit later in our video. But here you can see the notochord, and then above it, you have that infolding, right, to form that neural tube. Okay. Also, you need to know on either side of that notochord that's part of the mesodermal tissue, you have these somites that can begin to develop. And so I gave you this on your notes, midline mesodermal tissue that doesn't become the notochord. It's on either side. And these somites will become muscles. You want to put that in your notes, muscles associated with the axial skeleton. So axial is your center line and appendicular is arms and legs, okay? Axial skeleton, and this causes segmentation. Segmentation. Now, I need you to know, look right here. This is an example of differential gene expression. If it's limb skeletal muscle that you're going to develop right here, it's the PAX7, okay? But if it's body wall skeletal muscle, it's PAX3 and PAX7 in order to get that. So different genes are turned on in different places to cause cellular specialization. All right. So next, what we want to go through is the how. So now we know the what. So how does this all go down? So 3 big things you need to know: cellular differentiation, morphogenesis, and apoptosis. So on your notes, development requires 3 interconnected processes: cellular differentiation, which is cell specialization in structure and function; Morphogenesis produces shape and form, and I'll go through all of these, okay? Produces shape and form of the body and later pattern formation. And apoptosis, you already know what that is. Programmed cell what? Programmed cell death, right? Okay, so if you look at the rest of the notes, that's exactly, these are the last three things we need to go over. So specialization in the structure or function of a cell typically is caused by activation of specific genes. So some genes are turned on, others are not turned on. Now, that process of which genes are turned on or off has to do, remember, in my class, we've already discussed gene expression. We have talked about RNA polymerase's ability to bind to the promoter through the assistance of transcription factors, if you remember that. And there's enhancer regions and activators. There can be repressors involved, right? So that turning on of genes is going to be determined by what's in the cytoplasm of the cell. So when a sperm first fertilizes an egg and you're a diploid zygote, you are one cell, that cell is considered totipotent. And how long totipotency lasts depends on the species, depends on the organism. But this cell right here has full developmental potential. Now, after the first cleavage, after mitosis has been done one time, look and compare the upper cell and the bottom cell, okay? Which cell has more green proteins in it as opposed to red proteins, the top or the bottom? Right? Hopefully you can see the top cell, the ratio of green to red is higher, right? More green, less green here. Those green and red just represent some sort of protein. And then again, when you look at the next cleavage, the ratio of red to green is not the same. And this is what is going to act as transcription factors or gene activators to turn on genes in this cell is going to be different in the bottom right-hand cell because of the ratio of these two chemicals. Now, there's more than that, right? But that's the analogy that I want to use. And so it's the nuclear information, what's in each one of these cells' nucleus is the same, right? It came from that diploid zygote and you're doing mitosis, which is clones. But what's different is what is in the cytoplasm. So on cellular differentiation, every cell contains the exact same DNA and are totipotent at first, full developmental potential. So number one, and there's two mechanisms that lead to cellular differentiation. Number one, cytoplasmic segregation. And then I'm going to go through and give you an example here. Okay, so take a look. This is your zygote right here, okay? And maybe this is even before it is fertilized at this point. I want you to look at the different quadrants that we have. So first of all, you have the animal pole and the vegetal pole, not vegetable, not like carrots and tomatoes, vegetal. The vegetal pole is the pole that has the most yolk. Okay, and opposite of that is the animal pole. Okay, then you have another breakdown. The site where the sperm penetrates and then the opposite side of where the sperm penetrates. So the gray crescent will develop in frogs opposite of where the sperm penetrates. Now, this is what we know. If you touch the top of your head right now, that is where your father's sperm penetrated your mother's egg. Where that happens is your anterior region. Now, whether it's the front or the back of you has to do with yolk, okay? The front, okay, the ventral part here in the front is where the yolk is. Where there is no yolk will be the dorsal portion of you. That's where the animal pole is. So already we have four quadrants, right? It's sperm with yolk, sperm without yolk. And then the farthest part of you, your feet, the posterior end of you, right? Is it the front or the back of you has to do with the yolk or no yolk. And you can see that played out right here in our developing frog. So here's just one more picture to show you that. So you can see animal pole versus vegetal pole. Remember, vegetal pole has the yolk. Okay, and then here's your gray crescent that will be opposite of sperm penetration. So on your notes, you have all of that underneath the poles. I gave it to you, but go to cleavage. cytokinesis, right, will separate the ooplasmic, which means female proteins, the maternal determinants, which are okay, anyway, we'll get into more details there. The maternal determinants, and will activate different genes. So next, let's talk about an experiment that was done. So here is normal zygote. It undergoes cleavage, but instead of letting it develop, they took those two cells that had formed due to mitosis and they separated them. Okay, so that wouldn't normally happen, but they did it. They separated them. Both of them, because each had a piece of the gray crescent in it, developed into normal tadpoles. Okay, the second experiment right here, same thing, but they forced the cleavage so that one had gray crescent and one did not have gray crescent. And the one that had the gray crescent developed normally. The one without the gray crescent never got to the gastrula stage. It was paused at the blastula stage. So what does that tell you? It tells you that you need the gray crescent to go into the gastrula stage. So maybe you could think G and G, gray and gastrula, okay? Because for tadpoles, that's what they need to move to the next stage. And this just stopped at the blastula. It was never able to move on. Now, as far as totipotency, a few words I want to go over with you, and this is right underneath developmental potential there on your notes. So here you can see sperm fertilizing the egg. Okay, here's your morula. Remember, morula is a solid ball of cells, and at this point they are fully totipotent. Okay, then it will develop into a blastula, and then eventually this inner cell mass here will develop into different specialized tissues. So being totipotent, okay, that means you can give rise to all the cells of the body. Every cell you're going to make can come from a cell that is totipotent. It has full developmental potential. In fact, you and I, if you were to go in right after a sperm fertilized an egg and allow it to keep dividing, I think you can get to about 8 cells, and then you could separate those 8 cells, and all of them would develop into 8 identical octuplets, right? Would that be right? Because that's how long our totipotency lasts. For some organisms like mollusca, mollusk, clams, and things like that, they lose totipotency after the very first division. Like I said, that varies according to species. So totipotent, you have that one, full developmental potential. Now, the next two, okay, pluripotent, which is that inner cell mass, it can only give rise to the cells of the embryo. That's all that they can give rise to. So pluripotent gives rise to all the cells of the embryo. Okay, and then if you're unipotent, right, uni means one, okay, at this point, this is when you move so far along in development that you can only become that one thing. You have become determined and you will only differentiate into the one thing. So on your notes for unipotent can develop only along one line, Okay, so now we have talked about one of the 2 factors that influence development. So the first one was cytoplasmic segregation. So we already talked about that. Induction is the next one, and I've already introduced that to you, right? This is where one tissue influences another tissue. So how they're influencing that tissue is usually through transcription factors. Remember, transcription factors help RNA polymerase to bind to the promoter. So you can send a chemical signal, right, which activates some genes. Those genes are turned on. They're producing transcription factors, which then activate other genes, which can then make other signals, and et cetera, et cetera. And that was the chemistry we looked at behind the notochord and how it influences the ectoderm above it to fold in to form the spinal cord. So on induction for your notes, when one tissue influences the development of another, either by direct contact, okay, or by the release of chemicals and morphogens. And remember, cell signaling, we talked about this already. Cells are blind. So the only way you can communicate to another cell is through direct contact, like when we were looking at the interaction between a T cell and an infected body cell, that's direct contact. Or using like cytokines, right, to influence your immune response, or here through transcription factors. So it seems to activate certain genes, seems to activate these certain genes, okay, by generating transcription factors, So for instance, we know that the cells of the gray crescent in a frog are going to become, look at this folding, right? They're starting to fold in, right? And this will become the dorsal lip, the lip closer to the animal pole, right? The dorsal lip of the blastopore, this opening right here. Okay, so this would be the ventral lip down here, and up here is the dorsal lip of the blastopore, the opening. So you have that, and it's called, the gray crescent is called the primary organizer, and you have all of that. Okay, good. So moving on. This was an experiment done, and I kind of alluded to it earlier. So here they took some tissue that was supposed to be presumptive nervous tissue is what they removed. Presumptive nervous tissue. And remember, nervous tissue, right, comes from your ectoderm. So they took that and transplanted it onto another cell, but it never became a spinal cord. And the reason why it didn't become a spinal cord is that presumptive nervous tissue that was part of the ectoderm, you would have expected it to develop right there, did not have a notochord underneath it to influence it. So look at the second experiment. They took some presumptive notochord and they transplanted that. And when they did that, they ended up getting two nervous systems developing as a result of that. So that's showing you it's all about the mesoderm to get that specifically the notochord, to get the ectoderm to fold in. An example of induction, like what we discussed earlier. So on little II there underneath 2B, notochord induces brain development. Okay, now there's another great example. Remember I told you the lens of your eye comes from your ectoderm. So what you can see here, let me move myself over here. Okay, so there's something called your optic vesicle, and this is part of your mesodermal tissue, which causes the tissue right above it to fold in and become your lens of your eye. Okay, but then this is something kind of interesting. This this lens of your eye then gets this optic vesicle to become your optic cup and actually your retina at the back of your eye. So the vesicle induces the lens formation, and then the lens gets your optic vesicle to become a retina and a fully functioning eye. Now, they have done experiments on frogs. Again, I'm not saying that this is a good thing. I'm just saying it has happened, where they have moved multiple optical vesicles and put them along the legs of frogs, and they got eyes to develop along the legs of the frogs. No, they were not functional eyes because they didn't have the neuron migration to those to make those work. So that's why I also said formation of the lens. All right, let's keep going here. Oops. Okay, and so now let's talk about, I skipped a cell. I'm sorry, a slide. Here we go. C. elegans. So this is a nematode, which is a type of worm. It's a pseudocoelomated worm, and it has a delineated head. See how it comes to a point like the tail? They have studied this just to show you an example. This nematode is a total of 959 cells, and from the very beginning, they can predict which cells will develop from which area. And it takes about 3 days to develop. And I put that already in your notes. All right. So then the last big topic we need to discuss for today is morphogenesis. So morphogenesis is the movement. Remember, like even when we looked at the gastrula, right? Movement of cells that changes the shape and the form of body parts. So how does this all go down? So on your notes underneath, describe movement of cells that changes the shape and form. All right. And here, so morphogenesis is done by morphogens. That makes sense, right? These are chemicals or proteins that are in a gradient that will influence it. Now, let's think back to when I showed you anterior and posterior, right? And dorsal and ventral. Remember, the ventral part was where the yolk was, and the dorsal part was no yolk. And then we know here at this end, we have where the sperm would penetrate, and then the opposite is where there is no sperm penetration. But even before, in that egg, when the egg is forming, before it's even fertilized, there is variations in the amount of mRNA in different concentrations amongst that egg to begin with. And as a result of that, it starts to lead to what you can see here is that segmentation. So on your notes, go through morphogenesis, and we already have the movement. And then B says pattern formation, cells divide and differentiate that enable morphogenesis. And though there are so many different types of animals, they share common sets of genes that will direct this. So I want you to see in this picture is that you have different ratios of chemicals. So if you look right here at this end right here, you can see this GAP gene protein. So GAP gene codes for the GAP protein. It's concentrated here. And at the opposite end, you have a different protein. But look right here, you have a different one. You can see this is an overlap here, right? And you can see where the colors overlap. So this creates different ratios of chemicals within that developing embryo, which then can trigger these steps of induction, where one tissue is influencing another tissue. The reason why is it has different genes turned on in different locations depending on where it is in that embryo. Examples of abnormal development here is where you can, if you start messing with this, you can get double-winged flies, or you can have their legs coming out of their eyes, etc. Okay, so looking at these proteins that I just showed you in a ratio, developing anterior posterior, this is maternal determinants in the egg before the egg is even fertilized also, and this has to do with mRNAs. Remember, those are messenger RNAs that will be translated into proteins. And so here you can see you have your concentration of your hunchback, bicoid mRNAs over here. You have nanos and cotyl, and so they're all at different ratios. So just pick a point anywhere on here, and you're going to have rainbow different amounts of each of these. Just pick a different point and look to see the concentrations, and these different ratios are going to determine anterior, posterior, and ultimately segmentation. So on your notes, I gave you all of that. Okay, so here again, you can see nanos versus bicoid, and you can see this in a gradient. And on your notes, just so you know, underneath anterior, posterior, first step in pattern formation, results from maternal determinants, chemicals and morphogens in a gradient, and I told you bicoid is at the anterior and nanos at the posterior. Then you have what is called these gap genes, and the reason why they're called gap genes is because if you don't have them, you're going to have a gap in your development, and they're temporary, and they will activate certain regions in your body. So let me get you here, and here you can see that full segmentation or starting to get there. Here we go. Gap genes, these are for broad body regions, like think regions in your body, and they code for transcription factors, which will then activate some more specialized ones, which are called the pair rule genes. So on your notes, I have those for you. Pair rule genes, genes that code for preliminary segmentation, which in turn activate other genes, etc. Okay, what put this into place? Differences, remember, in the cytoplasm of that egg early on, right? It set everything into motion. And so here, reminder, morphogens are a chemical that gets morphogenesis to happen. So those morphogens can influence the DNA, and those morphogens could be chemicals in a gradient, the different ratios, which will then specifically, it's called a homeotic gene. And on your notes, homeotic genes are often called selector genes. I would highlight that because they select for segmental identity, and it's dictating body parts. Okay, these genes, once turned on, are dictating, right? Unipotent. You are only going in one direction from here on out, dictating those body parts. And that homeotic gene will code for a protein, another morphogen, which will turn on another one's homeotic gene, et cetera, et cetera. And so these genes, okay, and let me go. I think I have another picture here I want to show you. Okay, so here it's showing you the DNA. Here's a variable sequence. But these Hox genes, okay, these Hox genes are highly conserved amongst organisms. So homeotic genes control for pattern formation. The homeotic genes all contain a homeobox gene, and that's for short, you call it a Hox gene, okay, which will then code for this homeodomain protein, which is part of a transcription factor. So these Hox genes are coding for those transcription factors. So this you can see highly conserved. It doesn't matter if you're looking at a fruit fly, okay, or a mouse. It's showing you the Hox genes, and look at the similarity in what genes are active and what they control as far as where the head region is, where the midline is. This is a conserved, and this shows, right, this common ancestor, because you see the same genes turning on and making the same proteins in development. All right, and I gave you that in there. All right, and then we've done cellular differentiation. We've talked about morphogenesis. So the next and final thing we need to talk about in development is apoptosis, and that's death, which seems ironic, right? But death is a part of development. So for instance, when we're looking at that, developing tadpole, for it to develop into a frog, this tail's got to die, and that is called apoptosis. That is cellular suicide, right? When you hold out your hand, when you were developing as an embryo, your hand was a paddle. You needed apoptosis to occur in order for you to have phalanges. So that is already in your notes, that you need that programmed cell death for things to move forward. And here you can see when it doesn't happen, when you don't have that cellular suicide in between those digits, what that's going to look like, and that is incomplete differentiation. All right, so then I have for you this analogy. It might work for you, it might not work for you, but imagine you're playing pool, all right? The goal is to get all the balls in the pockets, right? And that would represent development. Playing the game of pool, that is morphogenesis, the process of playing that pool game, right? Induction. So here your stick, I don't know what you call it, stick, right? Cue stick. It hits the white ball hits the other balls, trying to get them in the pocket, okay? So what I want you to think about is that stick is the inducing tissue, like the notochord, right? The cue ball is the morphogen that is influencing like the ectoderm to fold in, and those could be transcription factors because it's influencing those homeotic genes, and that would be the colored ball. So this is influencing that. And to talk about cytoplasmic segregation, that would be like the way these balls are set up originally is going to help determine which balls are going to get hit into which pockets. So that was just a little analogy. I hope that was helpful for you. And that is the end. And if you are one of my students, I'll see you in class.